When we talk about our genome, the DNA in our cells, we think about protein-coding genes that are stably transmitted from one generation to the next. But in fact, things are much more complicated. The genome is far more agitated than it appears. A major threat to genome integrity is carried by the DNA itself. These are the transposable elements, also called jumping genes, because they can jump from one genomic location to another.
Transposable elements make up around 50% of the human genome. And as you can imagine, there is a huge evolutionary pressure to silence transposons. Their mobilization is highly mutagenic and can be at the origin of various cancers or genetic diseases such as hemophilia.
Reproductive cells, so sperm and oocytes, are the only cells that transfer genetic material to the host-plane. In these cells, a very sophisticated mechanism based on tiny small RNA has evolved to perform the specific function of controlling T, and thus are the gatekeepers of genome integrity.
That being said, their ability to move around the genome may also be an evolutionary advantage, mainly because they allow a genetic variation when mobilized. So genetic conflicts is often framed in terms of an arms race.
The fundamental questions that drive our research are how so tiny small RNA can control transposable element activity and how heterochromatin regions can play a key role in this control.
To investigate these different questions, we use Drosophila melanogaster as a model organism.
Drosophila is a really amazing model,
Easy to use.
and at the forefront in the study of molecular mechanism and evolutionary dynamics of transposable elements.
Our domain of expertise is multiple: genetics, cellular and molecular biology, and bioinformatics. We deploy a combination of techniques such as electron confocal microscopy, next-generation sequencing, cell sorting, but also classical molecular biology techniques.
Our strength is that our team is made of different scientists with different skills and personalities, enriched by national and international collaborations.
We are devoted to passing on our knowledge and training the next generation of curious and rigorous scientists.
Together, we investigate, we define new understanding questions, and we try to find or develop a technical way to answer them. We learn from each other and we learn for life.
This is not an easy task. Perseverance is required every day.
But what could be more challenging and exciting than always having new things to discover?
If you want to take part to this incredible journey
And learn more about transposable elements.
Do not hesitate to contact us.